Okay, well, thank you for the introduction. So something that Billy also mentioned was that we need to figure out where these VOCs that we measure are coming from. So this is one of the things I emphasized on during my PhD, which I finished last year. So I'll tell you a little bit about the research I did back then. So generally, when we talk about axial breadth analysis, we do the following thing. We have a clinical study. We get some data, right? We use, for example, the receiver. We analyze this data by GCMS or any other type of device. We get a chromatogram for each patient, and then we create a model. That's what we've seen in these previous talks as well. We get an AUC, a positive predictive value, a negative predictive value, and then basically we publish it, and that's where it ends right now, because we're not far along in the whole research process yet. Right, so this continues. We start a new clinical trial again, we publish, and we start another clinical trial, which is fine, of course, but we do know that there are some issues with axial breadth that we need to solve and that we cannot solve with just doing clinical trials. So what I did for my PhD as well is make an inventory of all the papers that were published on MS-based axial breadth analysis in lung disease. So I have an overview of what you can expect in these studies. You can see that half of these studies were done for lung cancer, and some others were asthma, COPD, cystic fibrosis, and a few, for example, ventilator-associated pneumonia. And the majority used GCMS, also some other devices, but in general there's quite I think, a consensus about using GCMS as a tool for this. For me, interesting part and also the shocking part is the statistics and the validation. So what you see is the orange bar in the third one, which basically says that almost half of the studies that were done actually only used univariate statistics. The issue with univariate statistics is, especially if you use data like omics that has thousands of variables, is that you get a lot of false positives. And you don't want that, right? You don't want to publish something that isn't true. What you should do, if you want to use multivariate statistics, is use multiple testing correction. And only a small part of those papers actually did that. The right tool to use is actually multivariate statistics. And in which form you do that, yeah, it does matter, obviously, but multivariate statistics is, in general, the right way to go for this type of data. The most shocking part I felt when I did this review of all the papers was that more than half of the studies did not use any type of validation, which I think is really shocking, right? Why are we publishing things that we don't know are actually true? Especially if we then use univariate statistics, right? Part of those studies, so approximately 40%, I think, did use some type of internal validation. Half of those used leave one out or k-fold cross-validation. The issue with that is that it's also not representative of if you would use an external validation, which is what you want. Only 2 of the studies in those 88 studies actually use an external validation, an independent external validation, which means that you go to a different center, preferably at a different time point, and you measure samples there. You don't use them in the model, you only use them for validation. So out of those 88, only 2 of those used that. And probably those 2 are reproducible. The other ones, we don't know. So this leads to many concerns that we've already heard as well. For example, that most of the compounds that we measure can usually only be found in one study. There is some overlap, obviously. For example, out of these 12 compounds, you can see that 2 of them are found in 2 studies, which I think is also concerning because there is apparently no consensus, even if you're talking about the same disease. So some of these concerns originate from variation between individuals or within an individual. I think we can really reduce this variation by using standard operating procedures. But they have to be written, right, by the community. There has to be consensus about this. And using devices like the receiver can reduce the variation as well. About the reliability of the models, we should really use multivariate statistics to make sure that the VOCs that we find are actually the right ones. And then we need external validation to actually make sure that our study is reproducible. Even though we can maybe find VOCs that work in one clinic or in one study, that doesn't mean they will work in a different clinic or a different study. Yeah, so external validation is key, in my opinion. For the reliability of the VOCs, we can do all these kinds of fancy clinical studies, but in the end we need to know where they come from, right? So we need the biological knowledge. So that's what one of the studies I did and what I will discuss today. The hypothetical origin of VOCs, and I think this has been proven, sort of, is that when we have a lesion or a tumor in an organ, like the liver, this tumor then produces VOCs in a different way than all the tissue around it. These VOCs enter the bloodstream, go through the alveolus, and then are exhaled. The same works for the lung. VOCs are produced in a different way, and we exhale them. One other option is as well that you get some kind of infection. You get a lesion which produces VOCs, and you exhale them. So this is the hypothesis, at least. We can simulate this in vitro. Let's say we have a cell. Cells have basic metabolism, so that means that they produce VOCs on a regular basis. Let's say we make this cell sick, so we add some kind of toxin to induce a reaction. That looks like this. What happens, what we expect to happen, is that the profile of VOCs that this cell produces changes. So this concept we used in an in vitro study that I did, where we have cells that are exposed to a toxin, in this case benzoate-pyrene, which induces DNA damage and can be found in smoke, for example. We had a control group, obviously, and we used this setup. So what we have here is a cell culture incubator. We had tubing going in and out. We had a gas flask that was providing air, and we had thermal desorption tubes connected on the outside. Basically what happens, can you see my... No, you can see my cursor. Basically what happens is the air gets flown through the tubing into the cell culture flask, and because there is flow, it will exit on the other side and then go through the desorption tubes. So you can capture the VOCs. We then did the same as we would do in a clinical study. We analyzed it by GCMS, and then we created a model. And the results show that we can actually distinguish between cells that were exposed and cells that were not exposed, right? So we have the blue controls and the red exposure group. And these were the VOCs that we found. So we have octadecane, for example, which is thought to be a byproduct of oxidative stress. We have nonanol decanol, which are thought to be related to CYP450 activity. But instead of stopping there, we decided to also collect other data. So instead of just doing breathomics, we also collected metabolomics, intracellular metabolites. And we also checked the gene expression. We did it in the following way. So for breathomics, we selected the discriminatory VOCs, as I just showed you. We did the same for the metabolomics. And for the transcriptomics, we did it a little bit differently than you would usually do. Instead of looking at each gene specifically, we combined them into pathways, because those are what we're interested in, and used pathway expression, and then selected discriminatory pathways instead of genes. We then combined this data using canonical correlation analysis, but also looked at the individual correlations. So here's the result of the canonical correlation analysis. What you see basically is the correlation between two groups of data, right? So we have the VOCs on one side, we have the metabolites on the other side, and we tried to get a correlation. What we find here is a correlation coefficient of 0.97, which is obviously a very good correlation. On the right, you can see the contribution that each VOC or metabolite has on this correlation. You can, for example, see that tyrosine is involved, and that our unknown VOC, unfortunately, is in this case very involved. Unfortunately, we don't know what it is. For the metabolites and pathways, we did the same thing. You can again see quite a good correlation coefficient, and you can see that tyrosine, glutamate, and glutamine are involved in this correlation, and also some pathways are are involved. And for example, mTOR does not seem to really contribute to this correlation in this case. So when we look at the individual correlations, it gets really interesting. What we see is, for example, that myo-inositol is negatively correlated with nonanol and decanol. So that means maybe, it's an association, maybe if one goes up, the other one goes down. We did the same for the metabolites and the pathways, and here we can see really see some clustering going on. So we have myo-inositol again, which is positively correlated with PTEN, mTOR, and feeder. And we also can see that glutamine, for example, is positively correlated with several pathways, including I4, AHSP, and several others. So combining all this data is now key, right? So we have VOCs versus metabolites, we have metabolites versus pathways. But ideally, we want to connect those VOCs to the pathways through the metabolites. So I have two examples of this, decanol, nonanol, and octadecane. So what you see here is a pathway from Biocarda. That's one of the pathways we had. We had a pathway expression for this pathway, showing that it went up after exposure to benzoapyrene. This was positively correlated to myo-inositol, which is a glucose isomer and involved in cell signaling proliferation. And since it can also inhibit the AKT pathway, which is in this pathway, we think it might be a compensation mechanism. If this pathway goes up, myo-inositol goes up and then inhibits the pathway. Myo-inositol itself is negatively correlated to decanol and nonanol. So it might be that we can say, it's an association still, but still, we can say that decanol and nonanol may be targets or may be markers of these apoptosis or proliferation pathways. We did the same for this pathway. So again, it's a Biocarda pathway, inhibition of cellular proliferation by Gleevec. This one goes down after benzoapyrene exposure and is negatively correlated with myo-inositol. Since it stimulates cell growth and survival, it's probably this compensation mechanism again. But the most interesting thing, I think, is the glutamine. So it's positively correlated with glutamine, which means that glutamine also goes down after exposure. And this one is very important because it's involved in biosynthesis of lipids and proteins. It's a very important metabolite in the cell. And it's been shown that its uptake into the cell is reduced after DNA damage. And obviously, when we add benzoapyrene to the cell, we get a lot of DNA damage. So this makes perfect sense in my opinion. Glutamine is negatively correlated with octadecane, so that means that octadecane goes up after exposure. And we've seen already that this is a byproduct of oxidative stress, probably also DNA damage. So we can probably, maybe, I should say maybe, use octadecane as a measure of oxidative stress or DNA damage. So these are two examples. 5 minutes? So this is the entire, I just showed you two examples, right? But this is the entire network that we made, where we have the underlying processes, so the gene expression pathways at the bottom. They are connected to the metabolites, and those metabolites are then connected to the VOCs that we found. You could see this as sort of the ingredients to make lovely scones, as we do in the UK, right? And the VOCs would be the smell of those lovely scones. That's the idea, at least. In conclusion, I do have to say this disclaimer, right? This is a hypothesis-generating study. This means that it's association, not causation. So what we found does not mean it's actually true. We need everyone here and all the other researchers in the exhaled breath research world to actually validate these targets. But at least we have an idea of where to look, right? So we found several VOCs that are interesting as viable targets for potential further research. For example, octadecane, nonanol, decanol. I think the next step would be to actually, for example, shut down these pathways or upregulate these pathways and see if these VOCs that we found go up or down, right? That would be semi-causal link. And this would allow us, as some people have already said, to actually narrow down the VOCs that we target, that we look for when we do a clinical test, to make sure that what we actually measure is actually biologically relevant. So I'd like to leave it at that. I think there's enough food for thought. These were my promoter and co-promoters during my PhD, and they were also very involved in this study, so I'd like to acknowledge them as well. That's it. Thank you.
Thank you very much. Thank you for that presentation. I'm sure there will be quite a few questions from the audience.
I'm Pierre Stefanito. I'm from the University of Liège, not so far from Maastricht. I'm an analytical chemist, and I have a question because with all the deep work that you do on the review, did you also highlight the study that did or did not perform chemical validation? Because everyone is mentioning like validation of the statistic, and I totally agree with that, but I think that a lot of studies are also missing chemical validation. The GCMS is not a magic tool. Mm-hmm. If you say that it's octadecane, it can be like something else maybe. And I think that one of the problems of lack of overlap between compounds, as you show, it's maybe because we are trusting too much the output of the system on the identification point of view. Because when I see branch alkanes, we all see a lot of branch alkane in breast. GCMS cannot differentiate branch alkanes. They have the same fragmentation. There's 2,3,6 3-methyl-dodecane and 3,4,6 is the same fragmentation. And I think that's something that we are really missing is the validation of these compounds, because we are still missing some technology to do that. We don't have the analytical standard for all of the compounds. I think that it's also something to take into account, and I don't know if in all the studies that you see which proportion really confirm the identification with strong gold standard of chemistry.
Yeah, you're absolutely correct. So I didn't look into that with these 88 studies, but that is a very big part of it as well. Yeah, I agree. I do know that there is some type of a reporting standard, I think, for chemical identification of metabolites. I think we should all use it. I don't think anyone is using it right now.
I never found a study, that's why I'm asking
the
question.
Exactly. I definitely think we should use them. What we did, I mean, I haven't been in this department for a year now, but still. What we did back then was spike the compounds on the GCMS, but still then you have the branching issue. So you're absolutely correct. I think that is also a very important part of it. Yeah.
Thank you very much.
We have, just to add to that, we have actually done internally at Alstom a study looking at that. And with our initial review of the literature, we found one compound that had been structurally elucidated in the proper way. So you're right, that's a big issue that the field also must look at, because it obviously ties into your ability to think about the underlying biology. Any other questions at the point in time?
Hi, Stefano Tassini from Alstom. First of all, great presentation. What I would like to ask is a couple of quick questions. So first of all, you are talking about pathways and metabolites and VOCs. So what are your parameters to say that, to essentially associate the pathway with the metabolite expression and the VOCs? So how do you judge that a pathway is activated? And the second question is more general. So pathways, my understanding is you use transcriptomics analysis, so perhaps mRNA level, and you can see differences in activation of mRNA going up and down. So there are many studies showing that the expression of mRNA is not directly related with the expression of proteins. And another issue is that the fact that there are proteins expressed at a certain level doesn't mean necessarily that they are activated or essentially their target expression is working in the specific moment. So the activity of the protein is not easily translated to the quantity of that protein. So how do you compensate or consider this kind of problems that you have in association with if your pathway is activated and related in correlation with the metabolites and VOCs?
So first question is, how do you tell whether pathways are upregulated and downregulated, right?
How do you associate the pathway with the metabolite and the VOCs?
Oh, okay. So just to specify, the pathways that we actually use are the ones that we found using multivariate statistics, right? To distinguish between the two groups. The associations that we found, and you're correct that mRNA does not mean that proteins are activated. I think this is an issue for the entire transcriptomics world, right? What I can say is that metabolites are the result of protein expression, right? So I think that using metabolomics as a way to assess whether proteins are active, etc., is the right way to go. Does that answer your question, sort of?
Yeah, in part. I was on the first part. I was more asking about okay, you use the pathway that come out from I don't know, an ingenuity pathway analysis or something like that. It's like, for example, if you have 3 genes, 3 mRNAs that are upregulated within the whole pathway, is that enough to say that it's associated with the metabolite, or you want to have all of them?
What we did was, so we have the gene expression data, right? So we did microarrays. We used an established method, I don't remember the name actually, which basically uses all the expression values that are in a gene set, in this case a pathway, to create a sort of pathway expression value. So that takes into account the expression of all the genes in a pathway. So that means if half of the genes go up and the other half go down, we get like probably like a neutral pathway expression. So that means that it's not upregulated or downregulated after exposure. Does that answer your question?
Yep.
Okay.
One final question, Paul?
Oh, this is a really simple one, I hope, and non-controversial.
It
Probably
never-
not.
It never is when you see
I think these in vitro studies are at the vanguard of breath analysis, and anyone who's serious about breath analysis needs to be looking at pathways and mechanism. And I'm interested, you did talk about pH control or pH manipulation of your cultures or headspace, so I'm offering you the opportunity to tell us a bit about the pH there. Thanks.
Yeah, we did test the pH of the medium for every sample, and they were similar. I think they were around 7, probably. I'm not sure, actually. Should look it up. It's been a while for me, right? It's been like a year and a half. So, but yeah, we did look into it, and there weren't any changes between the samples. So that shouldn't have a very big influence.
Great. Thank you for the fantastic presentations, all the speakers, and the fruitful discussion.